It's not vlogging.
Yeah,
It's not live streaming. Whatever my YouTube channel is about. But I've noticed that, as a teacher, one of my time commitments is grading and lesson planning. But now there's this big time commitment of responding to fan mail. Totally. Okay. So 2 objectives here. The first one is to talk about the purpose of eukaryotic gene regulation. The second purpose, or second objective, is to talk about the different methods of eukaryotic gene regulation. Those are my 2 objectives. The purpose and the methods. Okay. So let's start with purpose. Okay, so the overall purpose here of eukaryotic gene regulation, one of these purposes we've seen already, which is energy efficiency. We'll keep on coming back to this, right? This idea of energy efficiency, which is gene expression requires ATP hydrolysis, the burning of energy, to occur. And since eukaryotes have lots of genes, it's wasteful to express all genes. All right, because only some genes are needed at a particular time. To give you an estimate, for humans, there are about 25,000 genes across the 23 chromosomes. Right? And we only express a portion of those genes, depending on what part of the body we're talking about, what cells we're talking about. So it's energy efficiency. Okay. The second purpose is this idea of differentiation, and we'll combine this with development. So differentiation and development kind of go hand in hand, because since eukaryotes have higher complexity, they are composed of many different cell types. Right, so like a muscle cell, a nerve cell, skin cell, that's what allows for this complexity. So as an organism develops, gene regulation turns genes on or off depending on the function of a cell, and we call that process differentiation. All right. So throughout an organism's development, starting from like one cell all the way to trillions of cells, certain cells within their DNA, certain genes get turned on or off depending on the function of that cell. And just to remember, big remember here, all cells, or I'll put an asterisk next to all, like 99.9%, all cells in a eukaryote have the same set of DNA, but not all cells use the same genes. All right, so it's not that some cells in your body don't have those genes, it's that in differentiation, some cells use some genes and other cells use different genes. An example here, like a skin cell, it has the same 23 chromosomes as a cell in your stomach. Same 23 chromosomes. However, the cells in your skin aren't going to use the same genes as the genes used by the stomach cells. The protein pepsin, which is an enzyme that digests things in your stomach, is a protein coded by a gene that's used by your stomach cell, but not by your skin cell. Right? Melanin would be an example of a gene in the skin that's used by your skin cells, but not by your stomach cell. Okay, so those are our two purposes: energy efficiency and differentiation/development. Are there any questions so far, or things you want me to clarify? Okay, can you turn to your neighbor real quick? Can you summarize those two purposes? And then we'll come back. It's possible, yeah. All right, let's come back together in 3, 2, and 1. So, what we're going to talk about here is we're going to talk about some of the different methods. So we know the purposes. We want to keep those in mind. But gene regulation can take different forms in eukaryotes. What's the primary form of gene regulation in prokaryotes again? Operons. When you hear gene regulation in prokaryotes, the major method is operons. Eukaryotes, they actually have like 3 or 4 different methods of regulation. So let's talk about methods. Okay. So remember, prokaryotes use operons. Eukaryotes do not. All right. So I'm going to tell you about 3. There's 3 major ones that we need to be familiar with of how genes get regulated. So method 1 is chromatin modification. Chromatin modification. Does anybody recall what that term chromatin refers to? It's not a chromatid, not sister chromatids. This is chromatin. Do you recall what chromatin refers to? It's the casing of the genes. Let's unpack that a little bit more. Nicholas, do you recall what chromatin is? Okay. So, one method that eukaryotes use to regulate gene expression is by modifying the chromatin. So they do that in two different ways. The first way is they do something called histone acetylation. Histone acetylation. The second way is they do something called histone methylation. Those two terms, acetylation and methylation, those are just chemical groups that get added to the histones. So when a histone is acetylated, an enzyme adds acetyl groups to histones, which ultimately loosens up the chromatin. It loosens up the chromatin. And the reason that's important is that when it loosens it up, this encourages gene expression because RNA polymerase can attach easier. There's more space. Alright, so it's like this enzyme comes in and adds an acetyl group that actually kind of pushes the histones apart and makes the chromatin loosen up a little bit. What do you think methylation does? It tightens, right? It tightens the chromatin by adding a methyl group to histones, which tightens chromatin. I'm not going to write that again, but we can all see that if we tighten the chromatin, it's going to discourage gene expression because RNA polymerase can't attach as easily. Okay. So what I do want to show you here, if you take a look at the screen. So what this looks like, on the right here, I've got methylation. On the left here, I have acetylation. Right? Now, what these circles are representing are the cluster of histones. And then we have the blue, which is the DNA. Right? Zoomed in on a chromosome. Notice, right, the histones on the acetylation, they're spread out further, right? That's loosened up the chromatin. On the right-hand side, methylation, it tightly packs them. Right? And those little A's or M's, those are the acetyl groups or the methyl groups. A term that we're going to see, we call it euchromatin. When it tightens, loosens up, right? Less gene expression. modifications. So epigenetic modifications, or chromatin modifications, the last thing we'll say here is that so, before we move on, turn to your neighbor real quick. Can you summarize what is epigenetic modification or chromatin modification? What's that? No. We're going to talk about two other examples of methods. All right, let's come back together. So, epigenetic modification, chromatin modification, can influence gene expression. Influence gene expression. So, let's talk about second method here. Second method is called combinatorial transcription factor control. Combinatorial transcription factor control. So, within eukaryotic DNA, there's a common structure of a gene. So within a eukaryotic DNA, it's a common structure. And that common structure, it's got a couple different elements. So the first element it's got is, let's start with what we're familiar with. So this right here is the coding region of our gene. All right, that is what actually gives the mRNA that gives some protein. All right, and we know that within that coding region, which is a sequence, it's a sequence of DNA, we have introns and we have exons, right? What's the difference between those two again? What's an exon do? Evan, do you recall what an exon does? So if I have this few sections right here, let's say these are exons. These are introns, right? Mingjia, do you recall what those differences are between introns and exons? Could you elaborate a little bit?
When you're translating, you have to copy introns.
Right.
Because they don't code any
Right, they don't code for the amino acids. The reason I had you elaborate, we're going to press into that idea of introns. But introns are not useless. There's actually a lot of, they're useless in terms of the protein, but there's a lot of regions that actually are involved in regulation. So we have in our mRNA after processing, ultimately, the exons are the only regions that lead to the protein sequence. Okay, so we have our coding region of our gene coding region. Okay, and then just like in prokaryotes, we have a promoter and a terminator. All right, where the RNA polymerase attaches and detaches. Okay, and then usually what we have is a long stretch of intron region, and then upstream we have our enhancer regions. Just a quick note, whenever I use the term upstream, I'm referring to the sequence that occurs before the gene. Whenever I use the term downstream, that's after the gene. Upstream and downstream. Okay, so what we need to know here is that, remember that our promoter, that's where RNA polymerase attaches, right? The enhancer regions, what's kind of crazy about these is that you actually have multiple sections here. You have multiple sections. Where different transcription factor proteins can attach to affect gene expression. And why that's important here is that what we have, these three different regions in the enhancer section, is this can allow for expression in different cell types and time. So you have transcription factors that can attach to this enhancer region to maybe turn on this gene expression in the skin cell, or turn on this gene expression in the liver or wherever we're talking about. Okay. So, what ultimately happens here is, let's take, for example, let's say a transcription factor attaches to one of those sections. So this right here is a transcription factor. It's a protein that attaches to DNA. And remember that RNA polymerase attaches to the promoter. And do you all remember that video that I showed you where the DNA bends back on itself? All right? So what happens here is that this protein will actually interact with the RNA polymerase and cause transcription to occur. So just going to draw a simplified version of this. This is our gene. This is our promoter. This is my RNA polymerase. And my enhancer. Okay. Now, since we're just focusing on that green section from here, right? If a transcription factor is bound to that portion, it'll actually interact and then activate transcription. So why this is called combinatorial transcription factor control is that what allows different genes to be expressed is that different cell types express different transcription factors, or TFs. That attach to specific enhancer regions. So if you have specific transcription factors attaching to specific enhancer regions, the presence of those transcription factors allow you to turn on specific genes by activating gene expression. And what's pretty cool about that enhancer region is that there's multiple sections where a transcription factor that's found in the liver or the skin or the stomach can all attach to that same region of DNA and cause expression of that gene in those tissues. Okay. I know I'm going fast, but that's combinatorial transcription factor control. Okay. There's one more. The last one here is called micro or silencing RNA. Micro or silencing mRNA, and it's abbreviated as miRNA or siRNA. Not mRNA. This is microRNA. So there are regions, kind of going back to what we were talking about earlier with introns, there are regions in the eukaryotic genome which don't code for proteins but code for what are known as non-coding RNAs or RNA molecules. All right, in other words, there's sequences that get transcribed into RNA, but they don't become proteins. Okay. Now we know a couple of different examples of this. All right, what's an example of an RNA molecule that does not become a protein that's involved in the central dogma? What's an example of an RNA molecule that does not become a protein that's involved in the central dogma? tRNA. Right? Remember tRNA transfer RNAs? Those are RNA molecules that are actually coded for by some of these sections of DNA. So examples include tRNA, rRNA, which actually helps make up ribosomes, transfer RNA, ribosomal RNA. But there are these sections that produce microRNAs and silencing RNAs. There's another type called PWA RNAs. We're not going to get into that, but you can read about it in your books tonight. So these short RNAs, we're going to focus on these two right here. If you think about a gene when it's expressed, let's take this to be a gene, right? When it is transcribed, right, it becomes mRNA and then becomes a protein. So where miRNAs come into play they are these short RNA molecules that are transcribed from the genome that are complementary to a particular portion of mRNA. Right? Because if you think about when a ribosome is going to attach here, right, to start translation, what will end up happening is that this microRNA will actually come in and attach to the mRNA. And so when this ribosome is translating, this microRNA, bless you, is going to block that process from occurring. And even more, what tends to come with this molecule is an enzyme. That degrades mRNA. So if you think about this, what's the microRNA is doing is bringing that enzyme over to the mRNA, attaching to it, destroying the mRNA, and therefore blocking translation from occurring. This idea is that there's, in addition to transcription factors and epigenetic modifications, a eukaryote can produce microRNAs to repress or silence gene expression after transcriptions occur. right? If you noticed here, right, this chromatin modification is regulation prior to transcription. Same thing here. Is that both options A and B, they work by preventing transcription from occurring. MicroRNAs, we call this post-transcriptional regulation. Because it's happening after transcription has occurred, but you're regulating gene expression by destroying the mRNA. And this actually is a big area of research, because when gene expression is out of whack, you can actually make microRNAs as a form of medication to repress gene expression that's gone awry. That's a big area of research, is the production of microRNAs or silencing RNAs. Okay. Are there any questions or things that you want me to elaborate on that I talked about?